Thank you very much for inviting me. I'm just very happy to be here, very happy to share my experience as well. I think I've been really interested in medicine for lots of years. I put 30 years, it's a bit more than that actually, but I didn't want to put more than 30 because I thought people would not come here to listen to a dinosaur speaking of something which is quite new. So that's why I put 30 years. I've been extremely lucky in my life. I put it up front. Very happy. I think I've seen almost everything what a doctor can do now, and doing that on both sides of the world. That means the dark side of what people are saying, which is industry, of dark side of defense. I find it a wonderful experience. I've been also very lucky to work on lots of different continents. So what I want to share with you is my trajectory. I'm not saying it's the right trajectory, it's one trajectory, but again, it's been very fulfilling, and I'm just very happy to be here now at McGill. As I put in my title, really all my career almost have been driven by my excitement for drug discovery and drug development. And I just want to make sure that we're talking the same thing here. So what do we mean by drug discovery and drug development? And I will take here an example to show the various steps which lead to a drug being approved. The first thing is you need to identify a disease. What is the disease you want to go after? Is it HIV? Is it COVID? Is it dyslipidemia, coronary artery disease, depression? You need to identify that. And there are lots of parameters in this decision in terms of size of the unmet need, importance of the unmet need, suffering, drugability, and all that stuff. Then you need to identify a target. So if you decide to go and try to prevent coronary artery disease, for instance, which is, as you know, the #1 killer in the world, you need to identify how you want to approach this problem and find a target here. In this particular case, the target is an enzyme involved in the synthesis of cholesterol. It's called HMG-CoA reductase, and this enzyme can be modulated and inhibited by some molecules which were tested first and validated at a preclinical level, and they came up industry with HMG-CoA reductase inhibitors, now called statins. These molecules, when they have shown to be effective in vitro and in animal models and be safe, can then be tested into humans, and this is called first time into man. And then these experiments have really as a goal to demonstrate that a molecule or a therapeutic is safe, well tolerated, and see how much you can go up to in terms of safety. So tolerability and safety. And then once this being demonstrated, into proof of concept. That means showing that this molecule, which has been shown to be well tolerated and safe in healthy volunteers mostly, is doing the job. In this particular case, for statins, showing that these molecules were really lowering cholesterol levels. And then when this is being shown in trials, going to phase 2B and phase 3 trials, and these are big trials in large number of patients to demonstrate using normally 2 different studies that you come up with the same outcome, same benefits if you want. And then one is demonstrated, you go into regulatory filing and try to get your drug registered and approved, and then you enter the market and you go into life cycle management. You know how long it takes to go from A to Z in average?
Years.
Who is saying 5 years, 10 years, 15 years, 20 years in average? 5 years? 10 years? 15 years? 20 years? Okay. Give them, I can tell you, in average, once a molecule has been into humans, it still takes 10 years to go to the market. So it's long. It's interesting because this is way beyond, in terms of timing, what politicians have in mind. They shoot for 4 years to be re-elected. Here we're shooting for 15 years. Can you imagine? So that requires actually some visionary thinking. How much does it cost? Do you know how much it costs to go from here to there? In average, 100 million, 500 million, a billion, 5 billion. You can pick. Who is for 100 million? Who is for 500 million? Who is for a billion? Who is for 5 billion? Okay, lots of money. We all agree on that. I can tell you that if we're looking at the money which has been invested by the top 15 pharmaceutical companies in the past 10 years, so 2012, 2021, the top 15 companies pharma invested exactly $1 trillion. $1 trillion from the top 15 pharmaceutical companies. Of course, that's the denominator. What is the numerator? That means how many drugs have been approved, 500. So you can just do a very simple calculation, cost 2 billion in average to put a drug to the market. And then it's a risky business. How many drugs which go into man here end up on the market? Which proportion? 1%, 5%, 10%, 50%? You can pick. Who is for 1%? Who is for 5? Who is for 10? Who is for 50? You're quite well educated. So yeah, 5% in average, 5%. 5%. So it's very risky, it's very expensive, and it requires some long-term thinking. That's not something that academia can do, essentially. And I don't want taxpayers' money to be spent with such risky endeavors. That's why we have pharmaceutical companies. And no drugs has seen the light of day yet, just in academia. So you can already imagine that there is here an opportunity for cross talks, because pharmaceutical companies cannot develop drugs without testing them into humans, and humans are usually patients coming to the hospital. So it's very simple in this very simple equation to see that there is room really for interactions. And I've been very lucky to spend time on both sides. So this is the summary of what I've been saying. Probably $2 billion to bring a drug to the market between 8 and 25 years. I've not seen any drugs except some very recent gene therapy stuff coming out in less than 5 years. And success rate 4 to 6% once a drug has been going into humans. So this is the landscape I've been working with. And now, if you don't mind, and again, I wouldn't do that if I had not been invited to do so, I will share my own experience. Again, take that, whatever you think. It's been one experience. I'm just sharing my experience. Very candid. I started medical school in Switzerland in 78, so it's a long time ago. Finished up and graduated in 1984, and immediately went into clinical training. I trained as an internist, so internal medicine. I'm board certified in internal medicine. I've spent all my years going through the ER, through the ICUs. I've done my job, essentially. Small hospitals, middle hospitals, large hospitals. It's been wonderful because it, I think, has given me the chance to see a broad range of patients, because internal medicine is really covering, at that time, cardiology, neurology, so essentially anything, except surgery. And so internists don't do surgery, but what they have is drugs. And so that was really, I think, has sparked my interest in drugs, because that is what we have as a tool to improve health of our patients. Rapidly, I decided also that I wanted to go to academia and get trained, essentially, as a researcher. I liked practicing medicine, I loved it. But I didn't want to only use the recipes from others. I wanted to be part of those who label and write the recipes. And so I was very fortunate enough to be accepted early in my career, actually, in this guy's lab. Hans Brunner was among the first investigators at Cornell University, originally in New York, to test ACE inhibitors. ACE inhibitors are now very widely used to treat hypertension and congestive heart failure. This guy was the first one to give captopril to humans. And then he moved to Lausanne and he opened up his lab and developed really an infrastructure to test any ACE inhibitor going to humans and to demonstrate safety and tolerability. And so I went to his lab, spent 2 years in his lab, and really learned about clinical trials. These are not prestigious publications, but they are decent publications. I work, for instance, really looking at what is the effect of these drugs on the renin-angiotensin system in healthy volunteers. I also looked at some safety issues when you give ACE inhibitors. So that was really my first 2 years, just being exposed to clinical trials, what it is to give a drug to patients. In this particular case, mostly healthy volunteers. Wanted also then to do some more basic research, being exposed to what it is to go deeper, and then was actually sent by Hans Brunner. He said, okay, there is a good place where you can go to Melbourne in Australia. So I went with my wife, we're just wed, and decided to go to Colin Johnston's lab at the University of Melbourne. Spent 2 years there. It was great, really hard work. That means my first exposure to basic lab, to bench, essentially working on rats. Published a bunch of stuff, not prestigious, but decent work that I was doing there. I mentioned Stephen Harrap there. I mentioned him not because he supervised me, but he gave me an idea, which actually you will see during this career have been quite interesting. Stephen Harrap in this lab, Colin's lab, was dedicated, we were all dedicated to hypertension, but Stephen was interested in baldness. And I thought, what? In baldness? That's really not an interesting trait that you can really think of. But he was saying, yeah, it's a risk factor for cardiovascular disease. So bald men have a higher risk of heart attack. And so he wanted to learn more about baldness. So I just remember that later in my life. But I found that was quite interesting. Came back to Switzerland after my first 2 years of postdoctoral fellowship there. It was a great time there with my wife. We had our first baby there. It was fun. Decided to come back to Switzerland, finish up my training actually in medicine. But we really wanted to go back to research, and it's been a learning really. I'm not sure if I had to redo my life, I would have gone again to Colin's lab in Melbourne. It was a good lab, but it was not outstanding in my opinion. It was a good lab, and that was my choice. I decided to go there. I wanted to go there. I take all responsibility for going there. I think if I had to redo it, I would probably go to a different lab. But again, I have full respect for Colin Johnston and for having me there. But I was doing clinics then, and I have to tell you this story. I was still wondering, will I go to research or will I go to just practice? And there is no problem. That means there's a need for good internists out there. And so I thought maybe I should stay in the clinic. And there was this woman who was a rep from a company called Park Davis. Park Davis was, we're talking early 90s, was coming up with one of the first statins. I talked to you about statins before. It's called atorvastatin. And they were testing this atorvastatin. It was showing promises, but not on the market at that time, of course. And this woman said, Dr. Muser, if you're interested in going back to research and you should go into cholesterol and genetics. And I said, oh, I hate both. I hate cholesterol because it's fat, it's grease, it's disgusting. And I hate genetics because it's too complicated. And eventually, really, we are thinking, we're talking the early days. Remember, the gene for Huntington had been cloned in 1989. 1991, first gene associated with Alzheimer's by a guy called Alan Roses at Duke University. So people were thinking and starting to see the power of genetics. I'm actually just taking this back much later, 1999, but that was from the head of the NIH Genetics Institute, now later the head of the NIH, and he was talking about the Human Genome Project. That means that was wild. Can you imagine sequencing the 4 billion or 3 billion base pairs of the human genome? When in the early 90s, it was really hard to clone the first gene. But eventually, the idea was really that it would lead to new gene therapies and new therapeutics. So we were starting to think about it back in the early 90s. And that time I had learned from my experience, and I said, okay, now I want to go again into research, but really get trained well, very rough. I will go to the best place in the world where they do genetics and lipids. And I decided to go to Dallas, Texas. At UT Southwestern Medical Center, contacted these two guys. They had Nobel Prize for their discovery of the LDL receptor, and asked them if I could come to their lab to learn genetics. And they said, yes, you can. So I decided to take my wife. We had a second child in Switzerland in the meanwhile, and went to Dallas, Texas. Worked actually directly, not with Mike and Brown and Joe Goldstein there, but with a woman called Helen Hart. Helen was a young faculty there, and she was a wonderful woman, full of energy, full of passion, and also early in her career, so she really wanted to build a lab. She's been extremely successful. You can see here, unfortunately the picture is not good, but in 2015 she recorded for the Journal of Clinical Investigation, which is a very good journal, one of these conversations with giants in medicine. This is a spectacular woman, and she taught me genetics. I knew nothing when I arrived there. She taught me genetics. She taught me about lipids. I knew nothing about lipids before. It was a crash course on genetics and lipids. It was tough, and she was a tough woman. She said, okay, you can come to my lab, but be aware we work 6 days a week. And I said, yeah, it's hard, but kids. She said, okay, 5.5. So she was tough, but she was absolutely wonderful. And she's now actually also on the advisory board here of the chair that I'm having. So I've kept contact with her, and I've visited her, and when I have a problem, I call her, and she's still mentoring me, because in research you always have doubts, and it's very healthy to have doubts. Like to have some person you can call and say, Helen, I have a problem, can you help me? And she would guide me. So it's incredibly useful. And so that was my first paper, so in basic research, really going into deep human molecular genetics. It was fun, hard, but really enjoyable. But at the end, I was well trained. I've been trained in clinical research, I've been trained in basic experimental research, and I've been trained in molecular research. And I don't think I can emphasize the importance of being well trained. That's, if I may say so, I don't like to give lessons, but this is one still lesson: be very well trained. It means we're going for a marathon. I don't know if you've been running a marathon. It's long. 42 kilometers is a long run, and everybody says that for 35 kilometers there is a wall. It's hard. The last 5 or so kilometers are hard. You better have good shoes and be well trained. 42 years, that's the duration of our professional career, 23 to 65, sort of. You better be well trained and have good shoes, and when you start. And so I was very lucky because I was well trained, and I had a solid experience, which gave me essentially the confidence that I could start my own lab. essentially, which is what I did. It's always good to have a buddy with us, somebody that you can talk to, that can exchange ideas, and that is the most important thing, that can tell you your idea sucks. Because if you don't have this type of reflection, you may do mistakes. This is the guy who was sort of my buddy there. We knew him actually when we were in kindergarten almost. So, and he's been very well trained. He went to Harvard, and so when we merged our lab together, that was very wonderful. We sort of did some stuff at the molecular level. He had discovered the gene, I mapped it properly. I did also some pharmacogenetic studies at that time in the late 90s. Again, staying in the fields of lipids and genetics, because that's where I had been well trained. I was still fascinated by industry at that time. I was also seeing that the industry wanted to go into genetics big time. And actually, Glaxo Wellcome at that time had hired this guy, Alan Rose. I mentioned his name before. Alan is the guy who discovered ApoE as being a risk gene for Alzheimer's. So as part of the audience here is neurosciences, so you may have probably heard about ApoE and Alzheimer's. This guy discovered ApoE4 and Alzheimer's. Actually, he was in competition with Jules Poirier here in Montreal for this type of discoveries. And this guy had been hired by Glaxo at that time to build up a research program with the assumption that what he had done for Alzheimer's, that in finding a new gene for a common disease, would work for any diseases. And so he started and got the money, big money, to initiate projects and recruiting patients with a variety of different diseases: depression, Alzheimer's, asthma, COPD, you name it, and coronary disease. But of course, what this guy needed is people on the ground to collect the patients and recruit and get the consent and get the DNA. And so, again, because I was so lucky to be at UT Southwestern, I've been hearing about these people. And even when I was postdocing there, I had the chance to build up my network. And so I heard about Alan Roses, and when I started my career as an independent investigator, I got in touch with Alan and said, Alan, I've been hearing that you moved to Glaxo. If you want, I can collect samples for you. And he said, okay, go for it. Here is a check of $2 million and go run. So I'm not saying it's easy, I'm just saying that it's possible. It was very hard. We were tasked to recruit 100 families with at least 2 people affected with coronary artery disease before age 50, and who had survived this event so that we can collect DNA and consent from them. You don't find them that easily. We screened the entire French-speaking part of Switzerland to get these 100, but we got them very rapidly, just because we were just well organized, and it was also helpful for me to be a doctor and have been practicing. So we designed this gene called Study with Alan, and we actually also built up our own research program on these 100 families that we had recruited. So we were part of this big international network, but also built up our own research project onto that. And that went really well, I must say. It was hard, and I've been surprised working harder during that time. The moment where you start your own life as a PI is a delicate time. It's tough. It's tough. Being young PI is tough. But here it's been very helpful because you get this big money from Glaxo, of course, lots of pressure to deliver, but we made it. And eventually, they asked me, why don't you come to Glaxo and run the genetics for cardiovascular and metabolic for the company. And that was a very difficult decision to make, of course. It was 2002. I had my 3 kids then. Telling my wife and my kids maybe we go back to the US was bit of a question. But eventually, it was thought that why not? That means I loved the drugs in pharmacology. And again, there is no drugs which is being made on the academic side. So I decided to jump the gate and went to Glaxo. Decided to go to Glaxo in 2002 to Philadelphia with my family. Wanted to spend there 2 or 3 years. Ended up spending actually 10 years at Glaxo. And one of the challenges which I faced was the mandate I got. My job description was find new genes for cardiovascular and metabolic diseases that would lead to new drug targets. And of course, we needed to have a large number of samples. I had collected some samples, but not enough really to fill up the mandate that I had received. And so I went to Alan, who went to the top head of R&D at Glaxo, and said, I need money. I need to build up a new resource so that we can collect data from a large number of patients. And he said, okay, go any place you can find these samples. I came here to Montreal, actually, asking if there is any study where they could get samples for us to study, for instance, the genetics of hypertension. There was not enough at the size that we needed. I went to Boston, Framingham, I went to all over the world, essentially, to try to find some. But people were saying, no, we don't share with industry anyway. So I said, okay, we've got some very good friends in Lausanne. Let's build up our own resource. And I asked the head of R&D, I need $3 million to start a new study in Lausanne, at my hometown, with my former buddy here, Gerard, whom you saw in the picture. And we decided to build up a population-based study of 6,000 people. We're talking 2003 here, so that was before the UK Biobank. It was one of the first population-based studies. And we wanted to really capture these new technologies, which were available starting in 2006, 2007, to analyze a large number of variants in the human genome. It's called genome-wide association studies. And so as soon as actually we recruited these 6,000 people on time and on budget, we actually did these studies in these genetic studies. And I want to show these because that was the first paper which came out of these efforts. We run one of these genes. So what you have here is essentially the human genome on the x-axis. On the y-axis, you have what is called minus log p-value. That means what is the degree of significance of each of these 1 million dots on this plot. There are 1 million dots on this plot. And every time you've got one of these towers, it shows a series of variants or SNPs which are associated significantly, for instance, 10 to minus 100, with here LDL cholesterol. And so that was important to find this famous target for statins. We found it, actually, and that was important because it was telling that this approach was revealing existing sort of positive control that was revealing drug targets. So that went into the Lancet, we went very quickly into it. But when we designed this study in Lausanne, I also remembered Stephen Harrell. And Stephen was working, as I told you before, on boldness. So I was saying to these people, to Gerard and these folks, don't forget to record if people are bold or not. And we recorded boldness, actually, for these 6,000 people. There is a scale for that, it's called Hamilton scale. And in silico, we just run our GWAS analysis and found that with the androgen receptor, that's known, that we found that the first genomic regions associated with boldness. That was really cool. When we found that, it's always very important in this type of analysis when you test a lot of hypotheses, essentially 1 million hypotheses, to have what is called a replication set. That means an independent data set which will confirm or not your observation. And I didn't have that when I was at Glaxo, but people were telling me, go in touch with a guy called Brent Richards. He's at McGill, and he may have a data set for you. So I didn't know this guy. I called him and said, Brent, you have baldness samples. Can you check if house sleep there is associated with baldness? And he immediately responded because he had the data. And 3 months later, we had this paper in Nature Genetics. He was the first author, I was the last author. That was my first love story, if you want, with McGill. And so it's a long story, which started again in 1988. We were very fortunate because that was a machine. We had built up these capabilities. It was very expensive to build that. Eventually, with the recruitment of the patients, the genetic analysis, cost Glaxo $10 million approximately, but it was a machine. And we actually found, for instance, the first ones to find a new gene for liver steatosis, so fatty liver, if you want. And so we were just cranking the machine, and it was just wonderful. Being also in industry gave me the chance to meet best experts in the world to do genetics. And one day the CEO of Glaxo called me and said, I want you to work with 23andMe. I met these guys, 23andMe, at one of these cocktails, where all these big shots meet together. And he told me, I've met this woman at Virginia, she's the CEO of 23andMe. I want to do a project with her. So figure out what you can do. And so I contacted this woman. As you probably know, she used to be the wife of Sergey Brin, who founded Google. And we sat down together. I went to California. We sat down together and we figured out, why don't we just replicate in 23andMe what we have found in the Lausanne study, very simply. So same criteria, because she had all these data. And eventually, because she had a larger sample size, she replicated what we had. But because the sample size is bigger, power is bigger, and so she could detect more, or we could detect collectively more regions in the genome associated with baldness. So that was all very cool. In 2007 and 2008, there was a change in management at Glaxo, and that is something that, for those who are going into industry, or interested in industry, something to expect. Yes, it's great to be into industry. They have big money. They gave me $10 million to do this study in Lausanne. But these two guys, which I mentioned before, actually were invited to leave the company, and there was a new management there. But the new managers actually invited me to stay. This guy, you may know him for those who are in HBHL, because he's on the board of HBHL. Paul Matthews is in charge of imaging at Biobank. But he was at Glaxo at that time, and Len Crowden was my manager and gave me this chance to be in charge of genetics across the company. So across disease area, which was great. So I spent 10 years. And at that time, in 2011, for various reasons, I had my 3 kids going to college, and Lausanne was calling me back, actually. Why don't you run a big diagnostic lab of 600 people? You've been in industry. I've been lucky enough to be, I think, well trained in management and these type of things. Industry takes care of that. And so I decided to accept this job and went back to Switzerland for 8 years. I wanted to do precision medicine there. And what was very clear from my years at Glaxo is that 2 things. Firstly, genetics has really the power to detect new drug targets. But second, we need to think of new ways to testing new drugs. What I've been showing you, the cost and duration are not acceptable. We need to find ways to shrink that. And one way to do that is to use genetics. And this is what now we call precision drug development, using people who carry specific variants or a specific genetic makeup and take them into the clinic. And so I had built with Gerard the color study, which was really an observation cohort. I started an intervention cohort. That means a cohort of patients that we can draw patients from to enroll in the clinical trial. It was hard work. Again, any type of these things is hard to launch. It's really a 747 that you're trying to get off the ground. So it requires lots of work, frustrations, and stuff. But eventually we got it off the ground. We called it the Lausanne Institutional Biobank. And we were actually the first ones, I think, almost in the world to build up these type of infrastructures. We actually also looked carefully at the participation rate. It was quite a new program in genomic medicine, and found that actually a lot of people, if they are properly informed, are willing to be part of precision drug development. It's interesting, and I'm not shy of sharing that, but there was suddenly a new management in the hospital, and they didn't like this project anymore. And so that was a bit unfortunate. So for me, either going against the tide, which was not very pleasant, but at that time, I was called by Brent Richards, but also some headhunters, saying at McGill there is a terrific opportunity to build up a new genomic medicine program. And so after lots of discussions, my 3 kids had just get out of college, I decided to do like a second postdoc or a third postdoc with my wife and call it to McGill. And so take the plans and the maps and everything we had done and the learnings we had made in Lausanne and import it here to build up this Canada Excellence Research Chair in genomic medicine. The idea was very simple here, exactly what I've been doing in Lausanne, with the mission that Sarah was saying before. Our mission is to use genetics and genomics to support drug discovery, including precision drug development. So the idea here is let's get patients from the hospitals here, the MUHC, and Ryan is very supportive of this type of thing. The JGH, the Jewish General Hospital, the MNR. Let's also use patients with rare diseases, but also cohorts, CLSA, Cartagena, and so forth. Let's do whole genome sequencing on them, or exome sequencing, that means deep genetic analysis. Do find people who carry some specific variants and take them into the clinic to support these different steps. So that's the strategy that we have delineated, which has been approved, and we were just up and running. And I was very excited. I came here in August 2019. Everything was ready to really start, and then a problem came up. This problem. That's a big problem. If you imagine what we wanted to do, having to face that is not pleasant. Of course, this problem was coming also with NATE. We needed to understand the pathophysiological basis of COVID. We needed to find new diagnostic markers of COVID severity and outcome. That means really precision medicine at its best. But we also needed to find new therapies. And I remember that, and I'm sharing that with you again. It's all very personal stuff. But I was meeting the chief scientific officer of the province, Rémi Quirion, and the head of Genomic Québec, Daniel Collet, and the head of FRQS, le Fonds de Recherche du Québec en Santé, Carole Javet, to discuss this strategy and this project. And I remember my meeting, and it's hard to get an appointment with these folks who are very busy, was on the 19th of March 2020. And I got this meeting already 3 months earlier because, again, it's very tough to see them. And my idea was really to present my strategy, which I showed. And they were very polite when I met them, but they said, Vincent, we don't care. We don't care with your problem. We have another problem. We have a tsunami of problems coming up. Can you help us? And I said, yes, I can build up for you because I know how to do that, and I've done in my past life. I can build for you a biobank. That means really an infrastructure which will give researchers samples and data to do research and try to address these questions there. And so they said, what do you need? I said, I need your trust and your money. And they said, you can have both. And so it was wonderful, really. These guys took risk. Rémi Kirian, Carole Jabet, Daniel Coder took risk. They invested into these at risk. But told me, okay, build up your staff, take whoever you think in the province can help you. And so in a matter of 2 days, we assembled a team, a spectacular team, honestly, of people with expertise in genomics. Brent Richards was on board. Expertise in virology from the SHIR, expertise in law and ethics and communication and finance and you name it. I assembled this team with Carole Jabet in 2 days. And I think it's a Guinness Book record. We got the mandate 8 days after WHO declared COVID as a pandemic. 11 days later, not only the team was assembled, we had identified the question, the study design, the SOPs, we got the questionnaire in place, and started the recruitment at the JGH here at the Jewish Research. We recruited the first patient here. And in a month or so, a couple of months, we had 1,000 patients already recruited, which I think is quite spectacular. Now we're talking about 6,000 people who have been recruited. And whole genome sequencing on most of them. So a very deep phenotype. And that has been very instrumental for us because we could be part of large consortia trying to elucidate the genetic basis of COVID severity. For instance, this paper we contributed to Nature, where you find a series of peaks here, each corresponding to a gene which is involved in COVID severity. With Brent, again, really my partner in crime here, we characterized OAS1, which is on chromosome 12, and with Siri Zhu driving this analysis, really found these OAS1 as being protective for COVID. So it's been very exciting, very hard. Again, every time you want to build up something, it's very heavy lifting. We worked for this team of 10 experts, each with a different expertise, met every day, 7 days a week for 30 days, between 2 p.m. and 4 p.m. to just put together all that stuff. But eventually come up with something which would make sense, would deliver, and also would attract funding, because altogether we're talking really lots of millions of dollars to make it happen. COVID is not over, but with Brent, we're really wanting now to move to the next level, so capitalize on what we have learned from COVID biotech and move into the next set of diseases. We believe that diabetes is a big unmet medical need. There is lots of stuff to do. We are working with the MNI, that means the neuro as well, to see how we can collaborate and do really an impact now and use essentially what we've learned to make a dent in the genomics and precision medicine in diabetic complications. So what we really want to do is use genomic medicine again to do this type of research, but also apply that to the patient population at the Jewish, but also beyond that. So I think it's exciting. We managed actually to raise already first $1 million to start the project, and we are really hoping to get the first patients on board within this year. We are very sensitive as well on equity and diversity in genomic research. So we've organized, for instance, this symposium here on advancing equity in genetics and genomics in Canada. If there is any interest on your end, all that stuff is on YouTube now. So that's my journey. Sorry, I've been using the word I too much. I've been just talking about myself. But just want to share that with you very candidly. Thank all these people who have been funding and working with me there. I really want also to thank my wife, because she's accepted to do all these moves. When we came here, it was the 9th time we were moving transcontinental. So she's been very brave, and she has also really put her career on ice for me to develop. So I'm very grateful to her. Voilà, I'm done. If you have any